Hello, good day. My name is Okafor Uchesipre, a PhD candidate at the Faculty of Tropical Agri Sciences, Czech University of Life Sciences, Prague, on this scientific event in Bosnia on physiological response of tropical plant to abiotic stress. We may start by asking ourselves, what do you understand by stress itself? Stress is simply a deviation, a disturbance affecting plant growth and development. What are abiotic factors? Abiotic factors are simply non-living factors that affect plants. Plant physiology is the study of the functions of plants. So when something goes wrong with the internal functions, then you can say the plant is stressed. Tropical climates are warm climates. So in essence, tropical plants are plants that are adapted to warm conditions. Why do plants respond to abiotic stress? Plants, when the environment isn't conducive for plants, they try to adapt morphologically, physiologically, in order to survive. And there are some changes that are experienced within the plant and in the plant in order to survive. And this is why this research is very interesting because there are certain instruments we use to detect these changes in plants. This is an overview of how plants respond to abiotic stress. Morphologically, where you have reduced leaf area, which is peculiar to drought, cold especially, reduced growth rate. In fact, almost all abiotic stresses cause reduction in growth rate, decrease root to shoot ratio. This is evident in drought, where the root tend to, the root become longer in the quest for looking for water. When plants try to look for water, they go deep down the soil to look for water, thereby resulting in longer root to shoot. Then the physiological responses could be low photosynthetic rate, antioxidant accumulation, reduced chlorophyll content, stomata closure, and a host of others, which ultimately may lead to death if this stress persists. The molecular responses are expression of specific genes relating to stress. Now, in this figure, you see what I just mentioned, where the morphological, physiological, and molecular, how they all attribute to. So you can see the abiotic stress on the plant, and the plants respond to them. In the next slide, we I try to show the one of the essential instruments used to detect these responses, which is the infrared gas analyzer, which can be used to measure a whole lot of parameters, but not limited to stomata conductance, net photosynthetic rates, relative water content, and leaf transpiration. In this image, you can see the instrument handheld. It has a clip-like it has a clip-like stuff that holds plants, and the readings are obtained. So when a plant is stressed, certain values like the net photosynthetic rate reduces, while For non-stressed plants, they usually increased net photosynthetic rates. So this is a major instrument that is used, and in fact, when you want to embark on this research, this is one of the essential instrumentation needed. Then the next is the chlorophyll fluorescence. It's one of the fastest instrument you can use. This will give you a quick indication as whether the plant is stressed or not. Now I wrote here that this instrument is used to measure the chlorophyll fluorescence, which is the FV, the ratio of FV to FM. And I stated that when the value of FV FM exceeds zero point 0.7, it means the plant is non-stressed. But when it is below it, it shows that the plant is well stressed. Now the next slide, this is part of the work I was carrying out, where some values, as you can see, the the picture in the middle for sample 111 has an FVFM value of 0.728. This is a clear indication that the plant isn't stressed that much. And the next image just by the side for sample 56 shows the FVFM value of 0.644. Now, this experiment was conducted to know the effect of cold or drought. In fact, I carried out for both cold and drought. For cold, I had the treated plants were kept in a grow chamber at 5 degrees centigrade, while the control was kept at room temperature in the greenhouse. While for drought, we watered the ones, the non-treated ones were watered, while the stressed ones were deprived of water. We did not water them for some days to know how this plant will respond to them. And this data was obtained. We took the data as the experiment started. We took for 0 day, 0 hour, 24 hour, 36 hour, 48 hours, in that manner, trying to show how this plant responded. And we found out that this value, this FVFM, drops with time. It moved from around 0.8 to around 0.4. So that is just the indication what I tried to do. So this chlorophyll fluorescence using the OptiSciences OS 5P Plus is a quick indicator. It's an instrument that indicates whether a plant is stressed or not. And the picture with a plant and with a clip is just the process of trying to measure a plant's chlorophyll fluorescence, where a well expanded leaf is used for this data collection, and this a white clip is used to hold the plant for about 20 minutes. The idea is for the region of the region where the data ought to be collected should be kept in a dark condition for that period of time where this data is obtained from the leaf. And from this leaf, we could take about 3 readings, from the next leaf, 3 readings, and we can, from this plant, we can have up to 6 or 7 readings, and we can get the average from this plant. Now multiply by the number of replicates. Then the next slide is the growth chamber, also known as the phytotron, where this experiment is carried out. So as you can see, we have one of them where both drought or cold experiment can be obtained. One of the chamber can be used as control, while the other will be used as the treated plant. So the experiment was carried out in our faculty, a new faculty building, and so far so good. The experiment is going well, and other data is being obtained, like the pigments, where a section of the leaf will be taken for to determine the total chlorophyll, the total carotenoid, chlorophyll A and chlorophyll B determination. And we observed that with time, as the stress persisted, chlorophyll content was reducing drastically. So I hope this experiment is an interesting one, and I hope you all can join in this area of research to determine how plants respond to stress. Not limit it must not be a tropical plant, it could be temperate plants too. Plants in Czech Republic, we can also check how the frost can affect how frost affects this plant. So the study is really an interesting one, and I urge each and every one of you to borrow a leaf from my experiment and make science interesting. Thank you for your audience and have a beautiful day.